السلام عليكم جميعا. محاضرة رقم 11 كورس الجينيتكس لطلبة طب الأسنان. تمام. inborn error of metabolism حكينا شوية عنه. طرحنا إنه بالجينيتك فاريشن حكينا إنه بعض الناس يكون عندهم ميتابوليزم مختلف. في ناس عندهم أمراض بالميتابوليزم، مثلا حكينا فينيلكوتينيوريا. حكينا إنه ممكن بعض الناس الدراجز أو تعطيهم أدوية ممكن بعض يستفيد منه أكثر من غيره، ممكن واحد يستفيد أقل. طرحنا حكينا مثلا كيف إنه أدوية الكانسر بعض الناس ممكن تنفع معهم، بعض الناس ممكن ما تنفع. حكينا من الفاكتورز هي كان مثلا ممكن الميكروبيوم، بس other factors كمان اللي هم inborn errors of metabolism أو genetic variation of metabolism. هلا هون راح نحكي عن ال الابنورماليتي بالميتابوليزم، مش بس اللي كنا بنحكي عنه، بنحكي هون بس يعني exclusively عن الابنورماليتي في الميتابوليزم. genetic disease هل حكينا عن chromosomal disorders, numerical structure، حكينا single gene disorders caused by individual mutant gene. هلا وصلنا هون منهم راح يكون بعضهم يكون inborn errors of metabolism، وبعدين راح نحكي شوي عن multifactorial disorders. هلا إذا بتذكروا حكينا عن مثلا genetic risk factor, genetic protective factor، حكينا environmental risk and protective factors. كل هذول بيلعبوا دور في inborn errors of metabolism. هلا inborn error of metabolism definition of it is inherited metabolic diseases or congenital metabolic diseases. مع انهم كل واحدة لحالها، كل مرض او كل انزيم او كل substrate إلها مثلا مرض بكون it's very 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 rare. إذا إحنا بنجمعهم كلهم مع بعض بصير it's not that rare، بكون كلهم مع بعض تقريبا 1 إلى بين 1000 و 5000 incidents. هم it's a large group of genetic disorders resulting in a metabolic defect طبعا، which عادة بيكون single gene mutation، واحد الميوتيشن الجين اللي بيعمل الانزيم الفلاني اللي بيعمل ميتابوليزم للسبستريت الفلاني بصير فيه مشكلة. هذا البروتين مثلا بصير له ميوتيشن، بدل ما يكون في عندك مثلا الامينو اسيد الفلاني بيطلع امينو اسيد ثاني، او بيطلع مثلا ستوب كودون يعني ميسنس اور نونسنس ميوتيشن، مثلا بيؤدي الى تغيير عمل الانزيم. هذا الانزيم بطل يشتغل على السبستريت، بصير في عندك accumulation لهاي السبستريت، فبصير عندك هذا الجين برودكت انه نزل، بصير في عندك تبعات ل accumulation هاي السبستريت او accumulation لمثلا باي برودكت او مثلا toxic substance اللي كان مثلا هذا الجين البروتين اللي بيطلع من هذا الجين كان بيعمل لها detoxification او مثلا metabolism او catalysis او whatever. So هذه هي الفكرة من inborn errors of metabolism. هلا production of different protein with abnormal function مثلا cellular intoxication, energy deprivation, mixture of these illnesses. هلا metabolism as you know is either it's anabolic, you are building up, or catabolism or catabolic where you are breaking down substrate. Enzymes زي ما حكيت هلا قبل شوي play an important role in facilitating the process by serving as catalyst as you know من biochemistry in the conversion of one chemical to another. فتعرفوا هالحكي انتو مثلا انه انا if I replicate the DNA I get the RNA, I transcribe into RNA, I translate the RNA into the protein. The protein is usually this enzyme that carries out this function. فهاي هي النقطة. مثلا انتم بتعرفوا كتير بالبايوكيمستري بنبلش مثلا بكومباوند معين. you have الجين رقم 1، مثلا عندك هذا الكومباوند، عندك جين معين بيعمل لك انزيم، هذا الانزيم breaks down compound 1 into compound 2. Again you have another gene which controls another enzyme that breaks down compound 2 to compound 3. يعني النقطة هون انه مش جين واحد بكل عملية فيها كل الانزيم او كل الانزيم من جين واحد، لا، كل انزيم كل خطوة له جين معين طلع لك انزيم معين، طلع لك انزيم معين بتطلع لك انزيم معين وهو بيكون بيشتغل بخطوة معينة. فكل خطوة لها انزيم، كل انزيم له جين، فمعناته كل جين له mutation which will have its own تبعات معينة. مثلا إذا صار بالجين الأول في مشكلة، compound 1 ما راح يصير له compound 2، فيصير عندك تجمع للcompound 1 كتير مثلا. هذا التجمع compound 1 كتير شو راح يعمل لك؟ مثلا أعراض كمان. معناته إذا ما في شيء إنزيم 1 معناته compound 2 راح يصير كثير قليل. شو الأعراض اللي راح تطلع من قلة الكومباوند 2؟ فبتلاقي كل جين كل إنزيم له أعراض اليونيك له. ف this is the one gene one enzyme concept. Inborn errors of metabolism, a genetic disease also known as biochemical genetics. Gene level عندك gene mutation, protein level abnormal protein يا enzyme يا transport protein يا another protein which all have an abnormal metabolites at the metabolic level. تمام. Inborn error of metabolism is which there is a block at some point in the normal metabolic pathway, occurs due to the mutation in the DNA. DNA which codes for specific protein, enzyme, receptor, transport vehicle, membrane, pump, structural element. كل هذول مش ضروري بس enzyme. الفكرة من هاي السلايد إنه مش بس ضروري enzyme. receptors كمان اللي مثلا that capture a substrate or that transport the substrate or that actively transport a substrate that is needed, or مثلا a calcium molecule or a sodium molecule that is needed, or hydrogen that is needed for this specific area in the metabolic pathway كمان, or structural elements لنفس البروتينات. مثلا إذا بدك structure معين مثلا بيتا شيتس أو alpha helixes or whatever structure معين أنت بتحتاجه لهذا الإنزيم بالذات. كل هدول كمان بنضافوا على الفكرة هاي. هلا لما تيجي تشوف طلع مثلا التعقيد، مثلا بتعرفوا انتم اليوريك اسيد، عفوا اليوريا سايكل والالكترون ترانسبورت تشين. مثل كنا نحكي امبارح او المره الماضيه عن الالكترون ترانسبورت تشين، حكينا انه كيف انه في there's hundreds of proteins or tens of proteins. اشي من الجينوم، اشي من الميتوكوندريال الجينوم. كل واحد بضيف لك قطعة من البازل اللي بتعمل لك one protein مثلا اللي بيشتغل بالترانسبورت. This is in a way similar. There are missing defective enzymes necessary to metabolize the food eaten. Generally, they are autosomal recessive traits, and food not broken down properly may produce chemicals that build up طبعا in various parts of the body, causing medical problems and learning disorders. هذه هي in a nutshell what we are trying to say. فمثلا دخلت هالنيوترينز، عندك كل هالانزيمز بيستنوا بهالنيوترينز، عندك block in one area might be compensated by another mechanism مثلا، بس الcompensation هذا راح يصير له مثلا بضعف بقل مثلا او بكتر. ما عندك من هذا السبستريت بس عندك من هذا السبستريت، البقية الويست برودكت شغالة مثلا in this instance، بس there is مثلا a subtle change here that may produce excessive build-up of مثلا H or not enough P مثلا بهالحالة مثلا. So the end products of nutrition might not be as you would like it to be. وزي ما حكينا this is usually autosomal recessive traits. فبيسك idea حاط لك صورة منسف، عندك فياته المنسف فياته proteins، فياته sugars carbohydrates and فياته lipids, and you need factors to break all of them, and you need close interactions, and in the end if you have any deficiency, any problem, you might have a deficiency of one or an excess of the other. Both will have symptoms, not only excess will produce symptoms or deficiency alone. شو الأشياء المعينة ممكن يطلع منها؟ مثلا عندك بهالحالة مثلا أنت عندك glycogen, you are breaking down into intermediate substance tyrosine, you break it down to a simple molecule propionic A, which you break it down to get glucose, which you use as an energy substrate. هلا إذا أنت عندك الإنزيم الأولاني هنا صار فيه مشكلة, you will have an accumulation of glycogen. This leads to, if it accumulates, it leads to organomegaly and storage diseases. هذا هو glycogen storage diseases مثلا بهاي الحالة. إذا هذا الإنزيم ما في إشي بس عندك الإنزيم هذا بالخطوة الثانية في إشي، فصار عندك intermediate substance إلها accumulation. This accumulation of tyrosine is toxic وله toxic symptoms. ما صار مش المشكلة من هون، المشكلة بالبروبيونيك A. Again, its accumulation is toxic and has different, usually different or أحيانا mildly different symptoms than the accumulation of the other substance, the tyrosine. طيب ما عندك هذا المشكلة، بالنهاية أنت بتصير عندك deficiency في glucose which is an energy defect. فكل هذا again will have its own symptoms. فهنا نحكي general mechanism of problems. Substrate accumulates to toxic levels. This is if you are at the start of the chain. زي ما هلا كنا نحكي مثلا بالجلايكوجين كنا at the start of the chain. The substrate accumulates to toxic level. This is at the start of the chain. In the middle of the chain, toxic byproducts produced from shunting accumulated substrate. Middle of the chain is usually اللي اعطانا toxic byproducts. او in the end of the chain انت البرودكت تبع هاي التشين مثلا كان جلوكوز، اذا كان مش موجود you have deficiency of glucose, deficiency of the عادة end change end of the change of the chain بيعطيني انا something that is vital for the entire body. مثلا جلوكوز انا بحتاجه في كل مكان، فبصير عندك deficiency في عملية مهمة للجسم an end product that is necessary for lots of other مثلا cycles. فهون نطلع pure regulation results in overproduction of intermediates to toxic levels. Types: protein, carbohydrate or fatty acids. protein معمول amino acids, organic, urea cycle. ما بدي أعيد الحكي، ما راح نسأل هون أي شيء مثلا له دخل بالبايوكيمستري، بس إنه بدنا نعرف يعني as to refresh our minds. carbohydrate disorders: galactose, glucose, glycogen, fructose. fatty acids: medium chain, long chain, and so on. So mode of inheritance: these metabolic disorders are usually زي ما حكينا autosomal recessive. Consanguinity is always a relatively common problem. تزاوج الأقارب زي ما حكينا. And some are X-linked recessive conditions مثل Fabry's disease, ليشنيهان syndrome. ليشنيهان syndrome لازم نعرفها، يعني it's not that uncommon. يعني من هذول لازم يعني ليش نهان أقل ما في نعرفه Fabry's disease. A few inherited as autosomal dominant trait including porphyria, hyperlipidemia لازم نعرفها hyperlipidemia, hereditary angioedema, porphyria. هذول autosomal dominant طرحناهم حكينا عنهم لما كنا نحكي عن autosomal dominant diseases. فانتم شايفين هلا احنا خلال السنجل جين ديس اوردرز بس احنا بنعمم او بنخصص بالسنجل جين ديس اوردرز على الميتابوليك جينز. carbohydrate metabolism: galactosemia, glycogen storage diseases, pyruvate carboxylase deficiency, fructose 1,6 bisphosphatase deficiency, hereditary fructose intolerance, G6PD deficiency. Galactosemia results from a disturbance in the conversion of galactose to glucose. The enzyme deficiency causes an accumulation of galactose in body tissue. Classic type of lack galactose 1 uridyl transferase, GALT, galactose transferase or GALT. First 1 to 2 weeks of life عادة هذول they present with hypoglycemia with jaundice. إذا بتعرف هم بياكلوا كتير، أكلهم في عندهم جلاكتوز. الأطفال الصغار ما بيقدروا يحولوا ل جلوكوز، بصير عنده hypoglycemia، بصير في jaundice secondary to the intolerance of the galactose. You have galactokinase deficiency results from infantile cataract from accumulation of galacticol and galactose epimerase deficiency, which mostly confined to the blood cells. Estimated incidence is very low. هنا هذول هم glycogen storage diseases. خلينا نرجع لهم. metabolism of galactose بتعرفوه lactase بيعطيك galactose galactokinase. So in the same fashion if you have inability in the lactase مثلا you will not have the byproducts. In the end of chain is the glucose. So problem in glucose 1 phosphate هذا phosphate uridyl transferase, brain metal retardation, liver jaundice, hepatomegaly, cirrhosis, eyes, cataract. هذا كله out of the metabolism of galactose. Glycogen storage diseases في عدة enzymes اللي هون نرجع لهم هنا، بس إذا نطلع عليهم هنا many of the these, if you can see, a lot of them are storage diseases, and the storage organ عندنا إيش هو الليفر. بتلاقي كتير in these enzymes that the problem is in the liver or the muscle. Well, this is where you are storing these things: liver, muscle, liver, muscle, liver, muscle. This is the major affected tissues, are the tissues that store these substances or substrates. فاحنا ما بدنا نحفظ types 1 B 2 3 ما بدنا نحفظهم، بس بدنا نعرف انه the major affected tissue is the storage tissue. بدنا نعرف انه there is a lots of enzymes that are involved that are part sometimes of the debranching and the branching of the glycogen. carbohydrate disorders, classic galactosemia هلا بنحكي عنها، كنا نحكي عنها مثلا where the mutant gene. طبعا ما بدنا نحفظ الميوتنت جين شو هو والchromosomal location، بس بدنا نعرف it's a single gene disorder. One gene is affected here again. بعكس لما رحنا تحت شوي مثلا diabetes. Is diabetes a single gene disorder? No, it's a polygenic disorder. هذا metabolic disorder، وهدول برضه metabolic disorder، بس هذا تعرفوا diabetes كتير common، هدول are not very common. فهذا يعني حكينا من polygenic diseases، وهدول بيكونوا من single gene disorder which are much more rare، بس زي ما حكينا can affect can have an effect cumulative effect بينهم وبين بعضهم. glycogen storage diseases، فالفون كيرك والبومبي's disease are basically these two main diseases. So the glycogen straight chains, branch chains, and then you end up with glycogen as well, which you need to break down to get back the glucose after you store it. So the most common cause of glycogen storage diseases are types 1, 3, and 4. Type 1 اللي بدنا نعرفه الفون جيرك اللي هو the most common form of glycogen storage diseases. People with the type 1 don't have the enzyme that is needed to turn glycogen into glucose in the liver. معناته انت عندك الليفر فيها الجلايكوجين بتحاول تحوله لجلوكوز. Glycogen builds up in the liver زي ما حكينا in the storage area. Symptoms often appear in babies that are about 3 to 4 months. They may induce low blood sugar, hypoglycemia, and a swollen belly because of the enlarged liver. Type 3, people with type 3 don't have enough of an enzyme, the debranching enzyme that helps break down the glycogen. The glycogen can't fully break down, it collects in the liver and the muscle. Symptoms include a swollen belly, again from the enlarged liver, delayed growth, and weak muscles, لأنه هنا muscles are affected. Type 4 هذه الأشياء اللي نعرفهم يعني for the sake of knowing about these GSTs. Type 4 Anderson's disease عندهم هدول abnormal glycogen, particularly glycogen abnormal, which triggers body's infection fighting system, the immune system, which creates a scarring, cirrhosis of the liver and organs such as the muscle and the heart. هدول لازم نعرفهم الثلاثة. So فإذا عندنا type 1 is in the end chain نحول أنا glycogen to glucose. Type 2 type 3 كمان within the chain, the deep branching، فراح يكون عندي بالمسل بالليفر. Type 4 بالمسل بالليفر and the heart. Glucose G6PD سمعتوا عنه أدرستوا عنه بتعرفوه and you know that it's a problem when you favism سمعتوا عن favism كمان. So it's a problem, it's a metabolic problem. This is an X-linked defect. تذكروا حكينا this is an X-linked defect. It's an irreversible step of pentose phosphate pathway. Female heterozygotes may have symptoms. تعرفوا لأن heterozygous she has 2 X's may have symptoms, but the severity varies due to the non-random X chromosome inactivation. تذكروا كنا نحكي عن ionization. إذا عندها 2 X's, one of the X's is affected. فإذا أنا I am inactivating the one that is unaffected بطلع عندي خلية طبيعية. إذا I am inactivating the one that is not affected والaffected is هو being thrust forward يعني and is active راح يكون عندي هاي الخلية مش طبيعية. فبصير عندها هاي الهيتروزايجوس female عندها نص الخلايا طبيعي نص الخلايا مش طبيعي. فبكون ما بكون عندها symptoms بيكونوا may have some symptoms but it's not very severe. مين السيفير طبعا راح يكون ميل اللي عنده الطبيعي اللي عنده الاكس مش طبيعي راح يكون عنده severity أعلى. So the highest frequency is in the Mediterranean عندنا بمنطقتنا The hemolytic crises are usually in response to exogenous triggers such as certain drugs مثل antimalarial, food مثلا broad beans or certain infection بصير عندهم بحاجة بصير في effect على this enzyme. Lots of accumulation of G6 glucose 6 phosphate مش قاعدين بيقدروا يحولوه مثلا بصير في عندهم hemolytic crisis. disorders of amino acid metabolism بتعرفوا فين الكتونيوريا كتير بتسمعوا الكابتونيوريا هيموسيستينوريا oculocutaneous albinous and branched amino acids. بالبي كي يو بتعرفوا البي كي يو سمعتوا عنها it's a developmental delay in infancy neurological problems. فشايفين هلا احنا بنحكي عن لما نحكي عن الامينو اسيدز بلشنا نشوف هذيك بالكربوهيدرات كان في storage problems and energy problems. Here you will see it's a delay problems and it affects the neurons. فبصير عندك neurological manifestations such as seizures, mental retardation, behavioral problems. It's an autosomal recessive disorder. There's many mutations that are in this location that can lead to the same phenotype. Basic defect: mutation in the gene of PA hydroxylase and pathophysiology. PA or derivatives cause damage in the developing brain, and the treatment is to restrict this in the diet within 4 weeks of life to reduce this. So this is an inborn metabolic disorder. The first dietary restriction treatment, يعني this you can treat it with complete dietary restriction. The normal metabolism of phenylalanine عادة اللي هو شايفين انت pathway. So dietary sources you get phenylalanine هنا which you break into tyrosine or into body proteins and then it's broken down. This is not present, so you have an accumulation of this amino acid. So the metabolic pathway that breaks down phenylalanine and its relationship to certain genetic diseases. So شايفين إنه phenylalanine has more than one enzyme that is needed to break it down. So phenylalanine hydroxylase, phenylketonuria, you will get this with this enzyme. High levels cause progressive mental retardation, mental retardation, and the disease can result when phenylalanine accumulates to high levels. Hydroxyphenylpyruvate oxidase is tyrosinosis لأنه بصير عندك tyrosine is accumulates, so metabolic intermediates acid accumulates this intermediate اللي هو هنا, and it accumulates in the liver and prevents normal liver function. Alkaptonuria is homogenistic acid oxidase is homogenistic acid, and this occurs if it is not converted to malioacetic acid, and homogenic acid can accumulate in the tissues and cause premature joint and heart problem. هنا بنجي نوصل لمرحلة الجوتير تيست اللي هو كان one of the first mass testing for a genetic disorder in 1961. Screening of PKU was mandated in most states in the US at that time. Okay, alkaptonuria بدنا نعرفه this is autosomal recessive again. زي ما حكينا بنرجع ترجعوا للسلايد اللي قبليها. And what do you see? You have excreted in the urine dark color of the urine when it is exposed to the air. Dark pigment deposited in earwax, cartilage, and joints, and deposited in joint known as ochronosis and leads to arthritis. So this is normal urine. Urine from patients with alkaptonuria, you can see the darkening of the urine in these patients. Again, the main thing is the patients may display paleness, bluish darkening of outer ears, nose, and one of the eyes, and then long-term arthritis often occurs. فهاي نفس السلايد نرجع مرة ثانية. But these are for the amino acid disorders. اللي بدنا نعرفه phenylketonuria, what is the mutant gene product, alkaptonuria, homocystinuria. Urea cycle disorders. Urea cycle main function is to prevent the accumulation of nitrogen waste, as you know, which is waste is as urea. Uric cycle disorder responsible for the de novo arginine synthesis, and the urea cycle consists of 5 major biochemical reactions. ما بدنا نعرف نحكيهم، تعرفوهم من ال biochemistry, but as you can see most of them are autosomal recessive apart from ornithine transcarbamylase which is X-linked. فهذه اليوريا acid عفوا urea cycle disorders, you can have at each step you have one problem, and each step will have hyper accumulation of the substrate before that. Urea cycle defects, all but one of the disorders is autosomal recessive. Symptoms free period and then emesis, يعني بلش يحسنوا lethargy، بدأ يصير عندهم كوما. The features is طبعا شو رح يكون high ammonia لأنهم obviously they are not able to break it down, so they have high ammonia, low blood urea nitrogen, possible lactic acidosis, and absence of ketonuria. This is to differentiate it from a diabetic ketoacidosis. So ما عندهم ketone bodies موجودة، عندهم lactic acidosis, they are comatose بس ما في عندهم ketonuria, which is very important to differentiate it from diabetes. Normal to mildly low glucose، فهذول كمان راح يكونوا شو عندهم crisis، راح يكونوا جلوكوز كثير يعني مش normal. Treat high ammonia, infuse glucose طبعا, and use an ammonia scavenger مثلا biphenyl to treat this symptoms or this condition. So malformations may show episodic confusion and combativeness after high protein meals. Organic acidemia, acidotic with high anion gap. Urine ketones are high to normal ammonia. Often presents in the first days of life after dietary protein is introduced, and the baby has this drunk appearance. These are the organic acidemias and may have low white blood cells and platelets. You can check the serum amino acids, organic amino acids, or organic acids, and the urine to differentiate. So multiple carboxylase deficiency or defect in biotin utilization. Biotin is the vital cofactor of a vitamin, and its many pathways defects results in severe deterioration, dermatitis, alopecia, immune deficiency, and skin infections. So disorders of organic acids, يعني بس بدنا ناخذ فكرة يعني just a wide picture of them. Again, they're like the amino acid disorders, metabolic acidosis. They can have lots of acidosis, and so I don't want you to know the names of them, just to know that they are similar to amino acidemias as well. So organic acid disorders هذول بنعرفهم مثل malonic acidemia, propionic acidemia is very rare, and urea cycle defects بدنا نعرفهم هذول برضه. Lipid metabolism is the backbone of phospholipid and sphingolipids, biological membranes, and hormones. This is what you use lipids for. So intercellular messengers and energy substrates. So hyperlipidemia to the defect in lipid transport. Fatty acidemia is less common. Fatty acid mobilization from adipose tissue to cells, so energy substrate in the liver. Fatty acid transport across outer and inner mitochondrial membrane and entry into mitochondrial matrix. كل هذولا are defects where they can have problems, and defects in any of these can cause disease, short, medium, or long chain fatty acids. So LDL receptor تعرفوه عندكم pathway and regulation of cholesterol metabolism between ER to the Golgi, and then it is transported as or the transport of the LDL into the endosome. Mutation class between at synthesis 1 to transport to Golgi apparatus 2 للbinding على السيل membrane 3 للclustering اللي هو clustering of the receptors to the recycling in the endosome اللي هو type 5. هاي حسب ال LDL receptor pathway and regulation of cholesterol metabolism. The fatty acids are either it's long, medium, or short. Fatty acid oxidation defects, autosomal recessive inheritance. Again, medium, long, and very long chain autosomal diseases, and defects in acyl-CoA dehydrogenase, a mitochondrial um, protein, and is important in the fasting state. Key features هذول: an acute attack of life-threatening coma. هاي الكوما شو راح يكون فيها؟ hypoglycemia, absence of urine ketones again, and reducing substances. mild acidosis بالعكس هنا من الاسيدوسيس اللي كانت موجودة ما قبلها بالامينو اسيدز، بس هنا mild مع او بدون mild acidosis and hyperammonia. هناك كان باليوريك اسيد سايكل، باليوريا سايكل كان في عندنا الامونيا كثير عالية والاسيدوسيس موجود. هنا الاسيدوسيس مش موجود، بالهايبر امونيا مش عالية. مع هنا في عندنا hepatomegaly that might be also present. فهذا الاختلاف البسيط. Again, person will come, accumulates symptoms، يصير عنده acute attack with hypoglycemia، ما في urine ketones، في أو ما فيش بعض الأسيدوسيس and hyperammonemia. So diagnosis of serum acylcarnitine, mitochondrial transfer profile or fibroblast enzyme assay. فبيعمل هالassay you can tell from these the normal levels versus the abnormal. This is a normal profile, this is an abnormal profile. تطلع عليهم تعمل له إياه، يكون هذا النورمال يكون عنده هذا الاختلاف these different peaks. and abnormal you have a high accumulation of 1. طلعوا الأرقام كمان هون مختلفة، شايفين هون واصل 1.5 power 6، هون واصل ل 3. ففي there's a high spike of one of them. This can be diagnostic. So I'm not going to summarize the fatty acid metabolism. You know it, but this is the what we need to know is how the long and the medium chain are outside in the plasma, and in the cytoplasm you are breaking it down to in the end to ketones. Lysosomal storage diseases, prevalence is 1 to 5,000 to 8,000 live births, classified into 2 groups, enzymatic and non-enzymatic. Enzymatic deficiencies of acid hydrolases يعني من كل degradation of all these different types of molecules that you want to that we store, and we store them عادة they're usually stored in the lysosome, in glycogen, to polypeptides, to transport and glycogen, cholesterol, glycogen, glycan portion of the glycoproteins. Non-enzymatic, these are the enzymatic deficiencies, and then the non-enzymatic transport defects through the lysosomal membrane. يعني ما في مشكلة عندي بالenzyme breaking down of these products, but I cannot transport them. So both of them are storage diseases. هون الليسوزوم ضلها معلقة زي ما هي، شو ما كان فيها بس ما بتقدر transport الغاز. هون it does transport but has a problem in degradation of one of these substances, which leads to defective degradation. فبتشوفوا انتوا عندكم الاندوسايتوسيس، phagocytosis, phagosome. عندك this is the primary lysosome, secondary lysosome. You know all of this, but what happens is that when you are breaking down these products, if you have a problem in these lysosome, you will have an accumulation of one of these products. That's basically the idea here. Okay, let's skip this one. Lysosomal storage disorders. That's all what I just wanted you to know is from what we mentioned in this slide. Again, lipidosis diseases are many. Again, I think we can usually skip this one and go to sphingolipidosis. Tay-Sachs disease is an autosomal recessive disease. This is something that usually comes with eye and ear problems, developmental problems with blindness and deafness as well. Tay-Sachs is something that you might want to keep in your mind. So You should also skip this one. Okay. Okay. So this is the I don't know if we should go through this, but you know the lots of hormones that are made in the different uh, glands in the body where you can make these androgynous hormones, testosterone, progesterone to the female hormones, progesterone, and then the salt controlling hormones as well. Now, as these are also metabolic pathways, you know, I'm sure you have taken how each of these can be uh, affected by an enzyme. This is not a an exception. Aldosterone, testosterone, cortisol, progesterone, estrogen, كلهم هذولا are also part of the metabolic pathway. All of them are controlled by transport and degradation and building enzymes, which both will all have. فبتعرفوا إذا كان في عندك استروجين كتير شو راح يصير، إذا ما كان عندك تستوستيرون كفاية شو راح يصير، بتعرف إذا ما كان عندك الدستيرون كفاية شو راح يصير، كورتيزول again كفاية أو زيادة شو راح يصير. Again this one will also have. All of them are part of the cholesterol breakdown. فيعني this is what you need to know from disorders of salt loss to virilization. يكون مثلا موجود عندهم a female that has a lot of androgynous شو اسمه أعراض يعني androgynous، تكون عندها مثلا شعر كثير hirsutism مثلا deepening of voice or so on. You have taken this in many different areas in biochemistry and physiology and maybe pathology، بس هون بدنا نحكي عنهم نضيفها إنه cholesterol metabolism كل هدول كمان are still part of the inborn errors of metabolism. Testicular feminization, androgen receptor, female external genitalia, male internal genitalia, male chromosome، كل هدول مع إنهم مش part of the chromosomal disorders or sex chromosomal disorders, but this may give you sex problems, especially if it happens early on in the life. Between and then the salt losing as one. اللي بنعرفه 21 hydroxylase is the very most common salt losing steroid metabolism, and the testicular feminization problems with يعني لما يكون عندك testosterone in a female. The others البيورين pyrimidine metabolism ليش نهان حكينا this is an X-linked disorders لازم نعرفه as well. Heavy metal transport defects. What if you are trying to transport مثلا Wilson's disease? You know Wilson's disease. This is yet another metabolic disorders, genetic metabolic disorder. Wilson's disease is not that rare يعني 51,000 مقارنة مع البقية. Hemochromatosis for iron, you know this one as well. For summary, major inborn error metabolism presenting in the neonates as an acute encephalopathy بين urea cycle defects، راح يكون عندهم variable respiratory alkalosis, maple serum urine disease, metabolic acidosis with increased ion gap, non-ketonic hyperglycemia, no acid base disorders, organic acidemias اللي حكيناهم, metabolic acidosis with increased ion gap. هذولا this is the summary. Between the disorders involving complex molecule lysosomal disorders, disorders that give rise to intoxication, phenylketonuria, amino acid acidopathies, congenital urecycle بتشوفوها حكيناهم. This one we skip majorly, and then the sugar intolerances between galactosemia as حكيناها. Disorders that involve energy metabolism حكينا glycogenesis, glycogen storage diseases, and then we mentioned a little bit about the mitochondrial respiratory chain disorders. So inborn errors of metabolism associated with neonatal liver disease and later useful in the diagnosis. galactosemia شو بدك تعمل له؟ ممكن تشوف urine reducing substances, RBC galactose 1 phosphate transferase, tyrosinemia plasma constitutive amino acids نعملهم ونشوف التجمع للتيروسين neonatal hemochromatosis serum ferritin تقدر تعمل له and liver biopsy to see the hemochromatosis plasma for very long chain fatty acids skin biopsy for fibroblast cultures as well glycogen storage disease type 4 liver biopsy حكينا هذا اللي بيكون أكثر بالheart and then in the muscles as well and يعطيكم العافية هاي obviously this lecture we did not go into details on every اللي حكيناه بس هو المهم لأنه ما بدنا نعيد البايوكيمستري and physiology again we just want to have a major or a يعني a broad idea of these disorders. So again if you have any questions please send me your questions يعطيكم العافية.